In continuation to the previous lecture, today we will start a series of steps of mechanism of chymotrypsin. First, the target enters the active site of chymotrypsin and it is held there by hydrophobic interactions between exposed non-polar group of enzyme residues and the non-polar aromatic side chain of the substrate. It is important to note the hydrogen bond between the shift nitrogen on histidine 57 and the oxygen side chain of serine 195. Second, aided by the histidine-serine hydrogen bonding, the hydroxyl group on serine 195 performs a nucleophilic attack on the carbonyl carbon of an aromatic amino acid while simultaneously transferring the hydroxyl hydrogen to the histidine shift nitrogen. This attack pushes the pi carbonyl electrons onto the carbonyl oxygen, forming a short-lived intermediate consisting of a C-terminal carbon with 4 single bonds, an oxygen anion, the beta carbon of the aromatic amino acids, the N-terminus of the subsequent amino acid of the substrate protein, and the serine 195 side chain oxygen. Third, this intermediate is short-lived as the oxyanion electrons reform the pi bond with the C terminus of the aromatic amino acids. The bond between the carboxy terminus of the aromatic amino acids and the N-terminus of the subsequent residue is cleaved and its electrons are used to extract the hydrogen of the protonated shift nitrogen on histidine 57. The bond between the carbonyl carbon and the serine 195 oxygen remain in an ester configuration. This is called the acyl enzyme intermediate. The C-terminal side of the polypeptide is now free to dissociate from the active site of the enzyme. Fourth, water molecules are now able to enter and bind to active site through hydrogen bonding between the hydrogen atoms of water and the histidine 57 shift nitrogen. Fifth, the water oxygen now makes a nucleophilic attack on the carbonyl carbon of the acyl enzyme intermediate, pushing the carbonyl's pi electrons onto the carbonyl carbons as histidine 57 extracts one proton from water. This forms another quaternary carbon covalently bonded with serine, a hydroxyl, an oxyanion, and the aromatic amino acid. The proton on the recently protonated histidine 57 is now able to make a hydrogen bond with the serine oxygen. Sixth, the oxyanion electrons reform the carbonyl pi bond, cleaving the bond between the carbonyl carbon and the serine hydroxyl. The electrons in this bond are used by the serine oxygen to deprotonate the histidine shift nitrogen and reform the original enzyme. The substrate no longer has affinity for the active sites and it soon dissociates from the complex.